Alright, welcome everyone. In this video we're going to be looking at the structure of nucleotides, or the basic structure units when we talk about nucleic acids. If we think about DNA and RNA molecules, those are really composed of nucleotides that are linked to one another via a phosphodiester linkage to form long linear chains, right? That are then folded into compact structures. So we're going to take a look at the basic units. What are nucleotides composed of, right? Well, they're composed of a ribose, of a 5-membered pentose sugar. It's either a ribose or a deoxyribose, right? The ribose sugar has an alcohol right here, an alcohol right here. We have a methyl and an alcohol, right? My bad. So it's composed of a ribose sugar, or it could be a deoxyribose. And in a deoxy, it's really missing this alcohol group, right? So it could be either a ribose or a deoxyribose sugar. So a 5-membered sugar. It's also composed of a nitrogenous base that's either a purine or a pyrimidine derivative. That nitrogenous base is linked to this particular carbon atom, right? So when we're actually looking at the ribose or the deoxyribose sugars, we number them using 1', 2', 3', 4', 5'. All right, so in the deoxyribose, the deoxy lacks the 2' alcohol group. All right, and they're linked to a nitrogenous base, and at the 1' carbon, okay, and that nitrogenous base, I'll draw 1. Okay, that's an example of a purine. Nitrogenous bases are either purine or pyrimidine derivatives, right? A nitrogenous base, right? Those nitrogenous bases are either purine derivatives, and that's an example of a purine. Oopsie, yeah, that's fine. And this is really this double bond. They're either purine derivatives or they're pyrimidine derivatives. We have the bicyclic, and pyrimidines are all right. So that nitrogenous base could either be a purine or a pyrimidine derivatives. One nitrogenous base that we'll look at later is adenine, which really has a methyl group, right? So a methyl group. So it's a nitrogenous base. So if all we have is the sugar and the nitrogenous base, we call those two together a nucleoside. Right? A nucleoside. That particular pentose sugar could be linked to phosphate group at the 5' carbon, right? So, just gonna change the color of those bonds. Right. And we call this the alpha, beta, and gamma phosphate group. Right? The one closest to the pentose sugar is really our alpha our beta, and our gamma phosphate. Okay, and we call, in the presence of the phosphate groups, if we consider the entire thing, then we call those nucleotides, right? So nucleotides and nucleosides differ by, does it have the phosphate group or does it not, right? So then that is known as a nucleotide. It could be a nucleotide triphosphate, monophosphate, or diphosphate. All right, those specific bonds. Okay, so I meant to draw the oxygens, of course. Oxygen. The oxygen. All right, so now those bonds are known as a phosphodiester bond, right? I mean a phospho anhydride, a phospho anhydride bonds. Okay, those are the high energy bonds that basically are used, the hydrolysis of an ATP molecule that generates energy for coupling endergonic reactions is through the hydrolysis of those phosphoanhydride bonds. That specific initial bond is a phosphoester bond, right? That bond is a phosphoester bond. A phosphoester bond, right? Okay, so that's the basic of a nucleotide, right? We have the sugar. It's either a ribose in the presence of the alcohol group at the 2', or in the absence of this alcohol group, it is a deoxyribose sugar, right? That's the difference between an RNA and a DNA nucleotide. It's really the sugar that decides if it's an RNA or a DNA. If it's a deoxyribose sugar, then those are DNA, nucleotides part of a DNA molecule. And if it has the alcohol group, then it's part of an RNA, and it's connected to a nitrogenous base. They're either purine or pyrimidine. They're either purine or pyrimidine derivatives. If the nucleoside, it's called a nucleosides if it lacks the phosphate, and in the presence of 1, 2, or 3 phosphates, it's called a nucleotide. And the phosphate is linked to the 5' carbon, right? So we number the sugar using the 5', really, because when we number the nitrogenous bases, we don't use the prime, right? So this is really carbon 1, the first atom, nitrogen 1, 2, 3, 4, 5, and 6 for pyrimidine. In the case of purines, this is actually 1, this is 2, this is 3, this is 4, this is 5, 6, 7, 8, and 9. So purines are linked to the pentose sugar via the 9 atom, nitrogen 9, whereas pyrimidines are linked via nitrogen 1. That specific linkage is known as a glycosidic bond. And we specifically call it an N-glycosidic bond because the sugar is linked to a nitrogen atom, right? We could either have an O-glycosidic bond or an N-glycosidic bond, but in the case of nucleosides and nucleotides, it's an N-glycosidic bond. All right, so now we're going to take a look at the basic structure of the different nitrogenous bases that are either purine or pyrimidine derivatives. All right, and we could start with purines. Okay, one example would be adenine and really guanine. Those are our two. Adenine and guanine are our two purine derivatives, right? Adenine is simple because it's exactly like a purine. except it has a methyl group, right? There's a hydrogen here, but notice that's really later where the bond forms, but I'll keep the hydrogen. That's adenine. Okay, and in the case of guanine, okay, so a guanine has a carbonyl oxygen and an amine, right? The carbonyl oxygen is at this carbon, so now you get rid of the double bond because you have a double bond right here, which really means that you're going to add a hydrogen to this nitrogen if you got rid of the double bond, and it has an amine right here, which really means you keep this double bond. Okay, so that's guanine. Okay, we'll talk about some nomenclature later, but if adenine is part of a nucleoside, meaning if it's connected to a ribose or a deoxyribose sugar, instead of being called adenine, it's adenosine. Instead of guanine, it's guanosine. Okay, in the case of a pyrimidine, there's 3 different pyrimidines that we'll talk about. One is cytosine, right? Actually, let's talk about uracil and thymine first. Uracil and thymine, right? Uracil is only found in RNA molecules, whereas thymine is found in DNA molecules. They differ by a methyl. Cytosine. All right. In the case of uracil, so what we have is 2 carbonyl oxygens, one right here and one right here. Right? So I have to draw a hydrogen here and a hydrogen here. Uracil and thymine really differ by a single methyl group. So they're very similar, except that thymine, which is found in DNA, is methylated, right? There is a methyl right at 1, 2, 3, 4, 5 at carbon 5 of the nitrogenous base. Cytosine forms a bond. Later we'll talk about base pairing, but cytosine and guanine form base pairs. And think about that a purine is bicyclic, right? But one of the rings is really light, just like a pyrimidine. So when you draw it, and we'll just draw it as it is for a moment. All right, what you're doing is really swapping where the carbonyl oxygen is compared to the amine. So the amine is really right here, and the carbonyl oxygen is right here. All right, so that's cytosine. So we have adenine, guanine, uracil, thymine, and cytosine. Right? So that's the basic structure. So how about if it's forming, a dinucleotide? How are different nucleotides linked to one another? Right? So that's what we're going to take a look at. Next. Okay, so we're going to erase that. Again, the sugar and the nitrogenous base are linked via a glycosidic bond. We specifically call that an N-glycosidic bond because it's through the nitrogen atom. In the case of purines, it's the nitrogen 9. In the case of pyrimidines, it's nitrogen 1. All right. And to differentiate, we number the sugars using the prime system, right? So, and we have a phosphoester bond, and then we have the 2 phospho anhydride bonds, right, which provide a high energy source when like an ATP. So actually, let's talk about the naming for a second. So that's an example of a nucleotide. How do we name it? Well, adenine, if it's part of a nucleoside, instead of calling it adenine, we call it adenosine, right? And guanine, guanosine, right? So if we were to name this molecule, we would say adenosine. The phosphate is connected at the 5', so adenosine, 5', triphosphate, right? Phosphate. So adenosine is including the sugar, right? Without the sugar, just the nitrogenous base would be adenine. All right. And that's an example of ATP. All right, if we only have one phosphate group, we'll probably get to it in a later chapter, but those negatively charged oxygens typically will end up being complexed with ions, such as magnesium ions, to stabilize the charge, right? So later when we talk about ATP, we'll see in the presence of magnesium ions, and the role is really to stabilize the charge of those phosphate groups. Okay, so here's an oxygen. So instead of being triphosphate, now it is monophosphate, or AMP. Monophosphates, nucleotides have common names, and it ends in YLATE. Adenosine is adenylate, right? And that's true for monophosphate. Guanylate, right? And so forth. All right, so now how about if a dinucleotide is formed? Here's that. So different nucleotides are linked to one another by what we call a phosphodiester bond, and they're specifically linked through the 3' alcohol group of the nucleotide, right? And the 5' oxygen or phosphate group of the next nucleotide. So we are going to draw. Actually, for that name, yeah, that's fine. It was a deoxy ATP, right? Because it was missing the 2' alcohol group. Anyways, we have the sugar. We still have an alcohol group right here. And it could really be a nitrogenous base. We'll just draw one. Right? Suppose you have thymine. Okay. So they're linked. One nucleotide and another are linked through what we call a phosphodiester bond. Okay. So you have the phosphate that will link to this, right? So we call that particular bond a phosphodiester. You have the phosphate and you have the 2 esters, right? It's a phosphodiester bond, right? It's really important that you differentiate between the 2 bonds. If you know the structure, you know the name, right? That's a phosphodiester bond that links 2 nucleotides. And if you notice, it's the 3' alcohol group from one, and the next is at the 5', right? 3 to 5'. But when we're actually looking at amino acids or proteins, we're labeling them, or we're writing the sequence from the N terminus to the C terminus. And in the case of nucleic acids, it's from what we call the 5' end to the 3' end. So the 5' end is the one with the phosphate group. We call that the 5' end. And we write the sequence going towards the what we call the 3' end, right? The 3' end will always have this free alcohol group, right? So it's not part of a phosphodiester bond. Okay, and if we wanted to write the sequence of it, we're always looking at it from the 5' to the 3' end unless it was otherwise indicated. So what we have is adenine, right? But they're all deoxy, right? So we have deoxyadenine, and then what we have is thymine. Okay, so I hope this was helpful in terms of understanding the overall structure, being able to recognize the bonds, and being able to differentiate the names. And again, if you're learning the structure, remember one and let the other remind you of how it looks like. Okay, see you next time.